Art, I have been hanging for this one and I can't believe we're actually here doing it because I am fascinated about so many different things. But this caught my eye. And so I'm lucky enough tonight to be joined by Professor Andrew Pask, who is the head of Tigrr Labs. Now, I love the play on words that Tigrr Labs is. So do you want to explain that to people and a bit of your background, mate?
Yeah, so the Tiger stands for the Thylacine Integrated Genetic Research Restoration Lab, which is what we're really working on, is trying to bring the Tasmanian tiger back. It took me ages to come up with that acronym, but it had to spell something tiger, right? So I worked on that for a long time. But yeah, basically, I do a lot of conservation biology, particularly around Australian animals, marsupials in particular, and trying to think of creative ways that we can really conserve our species. And some of those ways involve actually, hopefully, bringing some species back that we've tragically lost.
I mean, firstly, congratulations on the acronym because it's just like perfect. But when we're talking about this de-extinction thought, it's amazing. Um, how did you get here? Like, is it something you coming out of high school and going to university, you ever imagined this is where you would end up?
I mean, I always loved Australian animals. I was obsessed with them ever since I was a little kid. I grew up in England. I was born there. We moved here when I was 10 years old. You know, I grew up in sort of inner London, and then we moved to a semi-rural area here in Australia. So as soon as I got here as a 10-year-old, it just blew my mind that there were kangaroos and possums and echidnas and all the things. So I was constantly like obsessed with going out, tracking animals, spotlighting animals at night, all of that stuff. Just really, really loved it. And then as I went through my schooling, I was always really good at biology because I really loved the animal side of things. I went to uni and did a degree in genetics. And I met a professor there who was working on marsupial genetics, trying to understand more about Australian animals. So it's super fascinating, their biology. And so I did a PhD there on doing basically just sequencing marsupial genomes, understanding about all of the DNA blueprints, how you get a marsupial, what makes them so unique. And from that was when I was really beginning to get super obsessed with the Tassie Tiger. So I was going into museum collections and getting specimens that we were doing work on all the time. And in the collections there, they had these amazing Tassie Tiger specimens. Like their pelts are just beautiful. Those skins are just absolutely incredible. They've got all the skeletons there as well. And I just got really interested in thinking about, could we learn more about the biology of an animal that had gone extinct? I was doing the work on animals that were still alive, but I thought, these specimens are not that old. They went extinct about 88 years ago. So most of the museum specimens are like 100 years old-ish. And so I really became fascinated in just going, could we get DNA from these specimens? This was back in the late 90s, early 2000s. So this is, before we had a human genome sequence. We didn't know that we could sequence whole genomes. We couldn't really do much in the way of manipulating DNA. People hadn't really done anything on really old DNA samples. This was sort of right back at the very beginning of that. And so I got some stuff from the museum. They let me sample some of the pelts, take little bits of material, and I was able to show that you could get DNA from it. And then because I was working on, amplifying genes and looking at genes and things from different marsupials, I amplified some genes. So a small little bit of DNA from the thylacine, I could actually get and read it and see what it looked like. And then from that, I was started asking questions like, well, if I can, look at it, can I actually see it function again? Can I bring it back to life? And again, we didn't really have good tools for doing that back in the day. So one of the things that we could do is put foreign bits of DNA into a different animal. So I took that little piece of DNA from the thylacine, from the Tassie Tiger, and I put it into a mouse and just asked the question, could I get that bit to work again? And it worked. So I could see that piece of DNA functioning and doing what it would have done in a Tassie Tiger. And that just blew my mind because it was the first time I think anybody in the world had demonstrated that you could actually get a piece of DNA from an extinct animal and actually make it function again. And then from there, I was hooked. I was like, well, if you can do it for one little bit, you can do it for the whole animal. This is just a matter of scale and being able to actually do that work. But back then, we just really didn't have the tools. But then, forward to today, 25 years later, and we do have the tools now to do it. And so it's something that my lab is very actively working on, is actually bringing that to reality, of actually bringing that species back.
Yeah, wow. I just hearing the foresight though, like, so this has been a long-term journey. So we're talking what, 25 plus years
Yeah.
with this vision. How does it
Yeah.
feel that so much progress has been made? Like, that must be such a cool feeling.
It's incredible. Like, I'm so excited about the fact that I've got a huge team of people now working on this project. I never got any funding for this, because you sound a bit like a crackpot, if you're going, I'm going to bring the Tassie Tiger back, and people don't even have like the beginning of the tools to do this stuff. So I'd been kind of chipping away at working on this. I always said it was my side hustle project. Like, this was the thing in the lab, didn't bring any money in, but I would constantly get these students who would come to me and they're like, I'm looking for a really interesting project. And I'd be like, hey, I've been working on this Tassie Tiger thing. And they'd be like, I love it. So I had this series of really passionate students who came through my lab, and they were all doing little bits of work on the puzzle of, trying to get this work up and running. And so we made some major achievements. Like, we sequenced the whole genome of the thylacine before we had any funding. We'd been really talking about how important it is to bring this species back and why we want to bring it back. And people, we were doing a lot of podcasts. I've been on the news a few times when we'd had major breakthroughs with this, because they found it really interesting. So I'd done a lot of outreach, talking to the public about this as well. And then all of a sudden, everybody just got super interested in this whole field of de-extinction science. There were a couple of big scientific books that came out about de-extinction and saying that we've got to start this conversation around it. And then we were just in this fortunate spot that I was probably one of the furthest along the road labs in the world that was working on trying to bring an animal back. And so, immediately we joined forces with a whole group of other labs with Colossal Biosciences. It's over in Dallas. And we just ended up getting some really serious manpower on this project of trying to bring this animal back. But to have that vision now, and all those students who came through my lab over that 25 years, most of them came back to work on this project. As soon as I got the funding for it, I'm like on the phone and calling them up and being like, we can do it. We can bring the thylacine back. And then they all came back to work in the lab again. So we've got just a great team of people who really love the Tassie Tiger and have been, passionately working on this project with me now for ages. So it's a really lucky thing. I'm a very fortunate scientist, and I love that we get to do this work.
I'm a massive animal lover, and the thought of having thylacines back on the landscape it's such an attractive thought to be able to consider that something that we wiped out, and that, I think unjustly the way we did it, and probably weren't, whether it be smart about it or, sort of the wrong information. And there was a whole aspect of it, but to be able to potentially bring that back, and I guess right or wrong, is so just absolutely fascinating that technology has got us to that point.
Yeah.
Have you had, like, I would think most people would agree with that. Do you get the other side that have sort of said, well, they went extinct for a reason, whether you like it or not, and they shouldn't come back? Do you ever get that?
Yeah, we do. I mean, I'm always happy to talk about that side of it as well, and people's opinions on that side. I mean, there's a lot of different de-extinction projects out there, like the Dodo, the Mammoth, the Thylacine. They're probably the big three ones bringing back the Tassie Tiger. I should say, I often call it the Thylacine as opposed to the Tassie Tiger. The Tassie Tiger, it's a great name for it, but it was found right throughout Australia, so it wasn't just Tasmanian. And it's a marsupial, so it's not a tiger. So the name could not be more wrong. But still, it's a good name. Like, it gets people excited about the animal, because it's, he's striped like a tiger. It had this incredibly beautiful pelt. But yeah, there are definitely a lot of people out there who just go, I think the biggest criticism that people have raised to me is always that there are so many species that we have in Australia that are on the brink of extinction, that aren't we better off spending this money, this time, this effort on saving those animals from going extinct, rather than spending this money on bringing back an animal that has already gone extinct? I think most people agree that, we shouldn't have hunted them to extinction, that this was definitely a mistake. And in fact, even at the time in Tasmania, they realised too late, right, that right when there was only one animal left in Hobart Zoo was when the Tasmanian government got together and said, actually, we need to pass a bill to protect this species. we've realised now it looks like they've disappeared from the landscape. And come to think of it, that's probably not a good thing. We probably need them in the landscape. And so they finally passed that conservation bill, but it was too late at that point. There was no
too little,
animals left.
too late.
Yeah, and that one animal died in captivity, and then that was it. It's one of the only species that we know the exact moment that species, ceased to exist on this planet. And it marks World Extinction Day to this day. It is because we know of that exact moment. So it's an absolutely tragic story. But, at the time, there were new European settlers in Tasmania. It looked an awful lot like the wolves of Europe that they had all contended with over the years. They were worried about their sheep. Sheep started going missing en masse. And so they decided economically it was better to go out and hunt this predator and to try and eradicate it than it was to deal with the productivity losses of their sheep. Turns out thylacines never ate sheep. That's really prey that is way too big for them. And, if you've ever been to Tasmania, there's plenty of small, medium-sized marsupials hopping around there that are literally just everywhere. And they're very easy meals for a thylacine. Whereas a sheep is actually quite an aggressive animal if an animal's attacking it. They're used to being prey species for things like wolves and foxes and other things. So they have a lot of defenses against them. Whereas our marsupials not so good at being prey. They tend to get eaten quite easily. So absolutely no way that, a thylacine's not in their diet. It's too big for it. But also it's a formidable animal for it to try and take down, as opposed to just eating the prey that they're used to. And then it turned out when they actually looked into it all, it was people stealing each other's sheep. There was a lot of sheep rustling that was going on at the time. And then they were, of course, just blaming the Tassie Tiger. So it is this absolutely tragic story of an animal that was misunderstood, persecuted for something it didn't do, unbelievably effective eradication scheme. So they offered, at the end of the 1900s, or the early 1900s rather, they were offering a £1 bounty for every thylacine that was brought in. And it actually was more lucrative for farmers to become bounty hunters for thylacines than it was to actually do farming. So there was a lot of people who just became professional thylacine hunters and were just out there just getting rid of them. One of the things is it's an apex predator. So it's one of those animals that sit right at the top of the food chain. They are absolutely critical for balancing out an environment and for making sure that all the other animals that sit beneath it are able to survive and thrive and all in harmony. And when you lose that apex predator, it really throws the entire landscape off. But as apex predators, you don't have a huge number of them. They're not animals that you would see, packs of thousands of them. They're quite sparse across the landscape because they eat a lot of animals, and so they need quite a big range in which they would normally cover. And so that means, unfortunately, it was super, super easy for people to go out and to just exterminate them. And it was, within the case of, maybe 20 years, they'd completely eradicated every single thylacine from the island of Tasmania. But getting back to that question, though, about people going, do we want to bring these animals back, and being against bringing these species back? I think if you understand the history of it and that real tragedy that you spoke about before, people go, it's very, very rare that I have people that still say, no, we definitely shouldn't do it. There's something morally wrong with this. People will say it's playing God. We shouldn't be messing around with these things. But the truth is, as humans, we mess around with these things in our environment all the time. We certainly messed around with the thylacine when we hunted them to extinction. We messed around with Tasmania when we cleared great swaths of land to farm sheep on it. Like, we're doing this when we make vaccines, everything. Everything we do is doing that. So this is at least doing something that's trying to right a wrong. But the reason for bringing it back isn't because thylacines are awesome, and they are awesome, and I do want to bring them back because they're awesome, but that they actually played that really important role in the landscape. They were so essential in balancing that ecosystem in Tasmania. And in the absence of the thylacine being there, we've already seen massive destabilisation of that ecosystem. The Tasmanian devil is nearly gone extinct. In fact, if humans hadn't intervened to save the Tasmanian devil from this weird disease that it gets, they absolutely would have been wiped out on Tasmania. They get this very strange facial tumour disease. It's a cancer, and that cancer is actually transmissible from animal to animal within that population. So these animals have these very large growths on their faces. Part of their natural behaviour when they're feeding and just in packs together is they'll bite each other's faces. Unfortunately, when they do that, if one of them has a tumour, it will spread to one of the other animals in that area. And so when you have an apex predator around, they actually remove those sick and wounded animals from the population. So if you have an animal that's sick, The thylacine swoops in, it eats that Tasmanian devil, and then it can't spread that disease anymore. Without the Tassie tiger there, without the thylacine, there is absolutely no other apex predatory mammal on that landscape. So there's nothing to kill those sick animals. So they literally persist in that population until the tumor becomes so big, most of them end up starving to death, or they have a hemorrhage, something awful, terrible death. And in that entire time that they're living in that population, they're just spreading that disease to everybody. So we know if you still had the Tassie tiger there, you wouldn't see that facial tumor disease, and it wouldn't have wiped out or nearly wiped out the Tasmanian devil. So it's a really important conservation reason that we want to put those animals back. So that question about does this take money away from conservation science, I always go, this is conservation science. It's a different way of thinking about it, but that thylacine, that animal back in that landscape in Tasmania, can do a job in preserving all of those other animal species in a way that we can never comprehend as humans. It's so complex, that interaction it has in that environment. But you've got to have those animals there. And apex predators are arguably the single most important part of that ecosystem. And we removed the only one that Tasmania had. So that's why I think, I can always argue to people that even if they think we're messing around with things we shouldn't, there's a really good reason in this case for this particular instance that you would definitely want to bring that animal back.
I mean, hindsight's a beautiful thing. And I agree with everything you just said, because I always talk about that we as humans have such an impact on the landscape, and whether it be playing God by doing, what you're doing, or just simply clearing land to make it more, whether we're going to farm it, whether we're going to house it, whatever it might be, we are essentially manipulating the environment to suit our needs, which has impacts. And exactly what you say, I talk about it all the time, that every population has a sustainable sort of limit. And when it goes over that, then you have that outbreak of disease and even the health of animals and starvation in different times and whatnot. So having that apex predator such as the thylacine, it's much needed. And you're exactly right with what's happening with the Tasmanian devil. And that's been a fascinating thing. And even watching that, having the, I think up around Newcastle way, they have a pen for them to sort of try and keep them going without that disease and flourish, so that if populations are wiped out, that they have backup, which is probably something we should have done with the thylacine. But hey, this story is going to be much cooler in the end, I feel. So how far is this away from actually happening? And then I want to sort of talk about the science behind how you're going to do this, because I think that's just unbelievable.
Yeah, so I mean, I can talk about both of those together, because one kind of leads to the other into the bit that's going to take a really long time, right? Because there are some bits that are relatively easy and some bits that are really, really hard. So the way that de-extinction science works is we still can't create life where there is none. So you can't take the tissues and the bits, the stuff we have in museum collections from thylacines, and somehow make that living tissue again. All the DNA has been shredded up into lots of tiny pieces. All the cells are completely dead. There's nothing you can really do with that. So what we do is you get DNA from your extinct species and you sequence its entire genome. So that's the complete set of DNA instructions that we have within every cell of our body. And that gives you that complete blueprint. That's what you need to know if you want to rebuild your thylacine or re-engineer it, basically, is exactly what that entire blueprint looked like. So that took us a long time. It was very, very difficult to do that just because of the quality of the specimens that we had. They're 100 years old. They're not in tip-top condition. They've been sitting in a shelf on the museum for a while. They're not great. They're not pristine. And so, that was a very big slog, but one of the major hurdles that we passed was actually having that full blueprint. Once you've got that, you then use that to look around in nature now and identify the closest living relative to your animal that went extinct. And so that for us is actually a really small mouse-sized marsupial. They're actually a bit smaller than your average mouse, and it's called a fat-tailed dunnart. So they're a little carnivorous marsupial, so they're a little carnivore as well. They eat a lot of insects and things, maybe a couple of small frogs and tadpoles and stuff, but they're pretty small little critters. And that is what we have now identified to be that closest living relative. Once you've identified that, you compare those two sets of DNA, the thylacine DNA, your dunnart DNA, and you identify everywhere that they're different. And like we are 98% identical to chimps, we share a lot of DNA with chimps. If you compare a thylacine and a dunnart, they're about 99% the same. So there's only 1% of the genome that makes a thylacine different from a dunnart. So basically when you know what that 1% is from lining those two sets of DNA up and seeing where those differences occur, then we can go in with our DNA editing tools into our living dunnart cells. So we take living cells now from our living dunnart, and we're just going to edit that 1% to turn those cells from a dunnart cell now into a thylacine cell. And once you've made that full 1% of changes, you've essentially re-engineered your thylacine. And then once you've got that cell, you can use standard cloning techniques. We do this a lot for livestock these days. We did it for Dolly the sheep many years ago now, but we have very good methods for turning that cell back into a whole living animal, which in this case would be a thylacine. And one of the super interesting things about this is that tiny little mouse-sized dunnart can be a surrogate mum, actually, as well as being the surrogate cell, if you like, for re-engineering the thylacine. And the reason this works is because marsupials give birth to such tiny, tiny babies that complete development in mum's pouch. And in the case of the thylacine and the fat-tailed dunnart, they're very closely related. Their babies are about the size of a grain of rice. So is the Tassie devil, actually. It's in that same group. So all of their marsupial babies are the smallest of any marsupial baby. Like, these things are absolutely tiny. So that means that even our little mouse-sized marsupial can actually give birth to a baby thylacine. It's obviously going to outgrow its mum's pouch and outgrow its mum very rapidly. But at that point, it's just suckling milk on a teat in the pouch. So we actually have artificial milk already for hand-rearing marsupials. When you find orphan marsupials or a killed mum that's got a pouch young, we actually have that for wildlife care. And so it would be a matter of then just hand-rearing those animals to independence. But getting back to the bit that's going to take a while and how long it's going to take, our genome, and it's this all mammal genome, so the same as a thylacine, is about 3 billion letters of code. So the thylacine's the same. It's got about 3 billion letters of code in its genome. So that 1% is a huge number of edits that have to make. So although we go like they're 99% the same, that 1% difference is a lot of differences. There's been incredible advances in the field in the last 10 years in the way that we can edit DNA. And that's what really made de-extinction science suddenly become a really feasible thing. It moved it from science fiction into science fact. Something we can actually do was this huge advance that we got in how well we can edit DNA. But it still takes a long time. So given our current state of where we're at with DNA editing, it would probably take about 10 years to make that 1% of edits. And we've made, a few thousand edits already, maybe about 50,000 edits. But we've still got a long way to go into making that full 1% of edits of our 3 billion base pair genome. But 10 years ago, this was unthinkable technology. Here we are today, and it's something that's very feasible, but could take a decade. And we're also working on improving those tools. So I'm really hopeful that in the next 5 to 10 years, there'll be major breakthroughs in how good we are at doing this editing and getting better and faster at it, that we actually will shorten that process down and we become much more efficient at making all of this DNA editing.
Just thinking about that, 3 billion and 1%, it's so much. And
yeah.
Does it ever get to that point where you just feels like overwhelmed that there's so much more to do?
There is so much more to do, but with every step that we do, we're making these major breakthroughs for conservation biology for marsupials, which I love. So while the end game for this is always getting a living, breathing thylacine, as we're going through this whole process, there's all these offshoot projects that we're working on where we can help save living marsupials that are on the brink of extinction right now using this same technology. So one of the great things we're able to do is use that same DNA editing technology to help save the Northern Quoll. So the Northern Quoll, they're these beautiful marsupials, spotted. They look like little Bambi of the marsupial world. They're about the size of a cat. They're just these beautiful little animals. And they are predicted to go extinct within the next 10 years because they're eating cane toads all over the northern part of Australia. Cane toads being a horrific introduced species into Australia. They're a toxic toad. None of our animals have evolved alongside toxic toads. And so any of our native species that eat these toads succumb to that toxin and die. So it's not just quolls. There's a whole bunch of other marsupials that eat them, but also our big snakes, our big lizards, all the goannas and things, they also are dying en masse from eating these toxic toads. They're just really an absolute scourge across Australia. But where these toads naturally occur, so in areas of the world where they actually come from, there are tons of mammals that evolved alongside those toads over hundreds of millions of years and have developed a resistance over all of that evolutionary time to the toxin of the toad, so they can actually eat them. And there are, again, tons of examples of mammals across all of those landscapes that can absolutely eat toxic toads and are perfectly fine. So when we looked into what it was exactly that was making those animals resistant, it's just one gene, so one little part of their genome, and just a couple of bits of code in that gene that you need to change to make an animal go from completely toxic to cane toads, then being completely resistant to that toxin. So you just have to change those two things, and an animal that would normally die from eating a cane toad is now completely resistant. So we got cells from a quoll, and we grew them up in the lab, and we made those DNA edits using the work that we'd done from the thylacine and figuring all that stuff out. And then we were able to expose those cells in the lab to the cane toad toxin. Normally those cells will instantly die from that toxin. And we were able to show that we can increase that toxin thousands of times higher than would kill the cells normally. And those cells are just absolutely fine. Wow. So we know now that we can just, go in and we can create this edit and we can save the quoll. This will definitely save the quoll from extinction. That's the major threat to them. It's not, habitat loss for those particular species. This is really, it's the cane toad that's the real pressure for that group of animals. So not only will it save the quoll, but then they can actually eat cane toads and help stop the spread of cane toads across Australia, which would be an amazing thing. So hopefully they can really bounce back numbers as well, that they've got this plentiful food source that suddenly they'll be able to eat. But this is a way of kind of taking that technology that we're developing now and applying it in a thoughtful way to save an animal that will go extinct if we don't step in and intervene. And although we're genetically engineering this animal, we are changing its DNA code. All we're really doing is speeding up that process of evolution. We know that this would happen if our quoll could survive another 100,000 years. It would probably have enough time to evolve naturally that resistance, because our DNA changes little by little over time, and we select for, the animals that would be resistant would be selected for, so they would get there in the end. But unfortunately, because we introduce cane toads and they've spread so quickly, quolls will be extinct. They will not have the amount of time they need to evolve. But we can go in and we can make that change, and then we can save the quoll from extinction. So there's great things that we're getting out of this project. So while that long editing project's happening and there is a lot to do, every single little step that we make along the way, we can immediately apply to some sort of conservation outcome for marsupials. And that's one example. There are many other things that we're working on as well that all have that sort of immediate benefit.
Well, I mean, that's an unreal repurpose of the project to be able to, do that quick adjustment. But that's a win-win, isn't it? For an animal to then be able to prey on cane toads, because that's what we lack in this country, and that they've been devastating. I'm a massive reptile lover, and seeing what they've done to the variety of goanna species and, as you said, snake species too, it's been heartbreaking, because populations that were massive are now really bare.
I love that you love the reptiles too, because this is the thing, right? It's easy to get public support for saving the quoll because everybody goes, the quoll's beautiful, and they are absolutely beautiful.
Little cute animal.
Real cute, fluffy, looks like Bambi. But the reptiles are, incredibly important in the ecosystems, but people just don't find them as cute. But it's that exact same strategy. So we would use this as just step one. Once everybody's happy with the quoll and we can get the quoll to go out there and be sort of an ambassador for understanding what we're doing, why we're doing it, how it works, the next thing would be to go out and start hitting all of our big lizards, all of the big snakes. It's that exact same edit that you have to make for any species. Doesn't matter if you're a mammal or a reptile, that will give you that resistance. And then hopefully we end up with our wildlife actually having a fighting chance against the cane toad and helping to stop the spread of that species, which would be just an unbelievable outcome. Yeah.
100%, 100%. So how does that work? So when you're doing the tweak to that DNA, are they passing that on? So if they're mating and then breeding, their offspring already have that new sequence? Or
yeah, so the way that this would work, so with some genes that you do this with, it's very hard to get that then to spread through a whole population of quolls and to get them to all be resistant. For this, I mean, it's an unfortunate thing, but because most of the quolls that won't have that DNA edit will die because they'll eat a cane toad at some point, this will be, like the only animals that are really very successful in the landscape and breeding well and passing their genetics on to the next generation will be these animals that carry that resistance gene. So this would be something that we would have to breed up a lot of quolls and release them, multiple times into the Australian environment to make sure that we get a really good number of animals that are carrying that specific resistance. But it will be selected for, so the offspring from those animals are going to be the only ones that survive to mate in the next generation. And so something like this will spread very quickly throughout the quolls, which is a really good thing. So it will enable that population to hopefully bounce back very, very quickly. And hopefully they figure out that they can eat cane toads pretty quickly as well. I mean, they certainly have a go at it now, which is what's leading to their death. And so once they figure out that they're fine, hopefully that will really help their populations bounce back again, because there is a very plentiful food source out there in the cane toads.
Yeah, 100%.
Yeah.
So back to the thylacine. So when we're talking about, you've got an 8-step, I think the project you said has 8 steps off the top of my head.
Yeah.
And the first 2 have been completed. You're in the mix of number 3. And
the DNA editing phase. Yeah.
Yeah. And then you're continuing.
Yeah.
Is there do you have KPIs that you have to hit or milestones in regards to each of these steps, or?
Yeah, I mean, we're trying to do this as quickly as we possibly can, because the longer that landscape is missing the thylacine, the more it changes in its absence. So we're really driven by that. But, we work with this big company who are really interested in trying to develop pipelines for de-extincting animals. And the big push of Colossal Biosciences is really trying to undo all of these mistakes that we've made as humans, is that there's been too many instances where we've realized too late that we've lost a really critical animal in the landscape. And it's going, can we go, beyond the technologies that we currently have to think about actually bringing back some of these species that we lost and going, maybe we can reverse some of those extinctions. And then that can help us re-engineer, if you like, some of these ecosystems so they become balanced again. And we can actually prevent this incredible mass extinction that we're seeing on our planet at the moment. We're losing species today at a rate that has never been recorded in our planet's history ever before. And that includes like the fossil record. When we've seen these previous mass extinctions, they occur over a much longer time frame than we're seeing driven by humans today. We're really driving this huge mass extinction. And the truth is, even if we were to stop global warming, habitat destruction, and any more extinctions tomorrow, for a lot of ecosystems, it's already too late. Like, we've gone too far. We've tipped over that tipping point of where things are going to continue to spiral down. We've reduced the size of populations of animals so much that it's really difficult for those to bounce back. But this is where all of those de-extinction technologies can come in, and it can be the only hope we have of reversing what we call this extinction vortex, when an animal population becomes so small that they really just can't thrive again. They've lost too much of that population diversity and they just can't really survive. This is really what we've seen happen to the Tasmanian devil. They've become such a small population now because of this disease. There's really no way to bounce them back. We've bred them on mainland here so that we can breed them in the absence of the disease. But if you put them back in Tassie where they belong, they're just going to pick up that tumour again, and all the animals are basically still doomed from that disease at the moment. But we can use de-extinction technology again to go back into museum collections, to get DNA from Tasmanian devils that lived 100, 150 years ago, and to re-engineer some of that lost diversity back into the animals today so that we can help those populations boom and thrive once again. But yeah, we're all working on KPIs and deadlines for this stuff. There's a bit of a race on. I think we're all pretty competitive about who's going to achieve de-extinction first. I think I'm fairly confident Thylacine is going to be the winner, but we have to wait and see.
So who are those competitors? I think you mentioned before it was the Woolly Mammoth and the Dodo are probably the other two main ones that are getting back.
They're the ones that are sort of, truly in the race.
Are they? Look, I mean, is that accurate with the Woolly Mammoth and bringing them back on the landscape? That sort of, I don't know. When I sort of hear, and the whole thing fascinates me, obviously.
Yeah.
But I just feel that the thylacine is the most important there to get back on the landscape.
Yeah.
And the Woolly Mammoth is a completely different animal. Like, I mean, it's been gone for so long compared to a thylacine, which in in extinction terms, very small amount of time.
Yeah. I mean, humans probably hunted mammoths to extinction. We feel fairly confident about that, but still it was a different time, right? Like, this was very primitive man, and they were, 10,000 hamburgers walking around. So they were an easy meal for them to get. And if you were bringing down a mammoth with a group of, humans, it would feed everybody for a long time, and they had amazing hides that you could make skin from and huge fat stores that you could use for oil and other stuff. So they were a very useful animal on the landscape for humans to be able to hunt and kill. And, it was probably part of our success was that we were able to hunt big animals like that would have, really helped those populations thrive. But they have been gone for a long time. This is thousands of years. The world has changed quite a lot since those animals have gone. But there's a lot of really important evidence about, what these animals did in the landscape. Mammoths were really great ecosystem engineers. So they would clear very large areas of forest. They were quite destructive. So they would smash down a lot of trees. They did this particularly in the tundra. So in these, really sort of cold and parts of our planet. And when they do that, there was a lot more meadows that would grow in those regions. Within the absence of trees, you get all the grasses and stuff growing. Grass is actually really amazing at sequestering carbon, so it's actually a very good carbon sink, better, more effective than the forests. And also, when that land was open, it actually cooled down a lot more in winter, and so you got a much deeper area of permafrost, so a much deeper frozen part of our planet that stayed cold for longer when we moved into summer phases in the different hemispheres. And so that area of frozen ground could actually, if we were able, because we've lost the permafrost, there are many forests across the tundra now, the globe's getting warmer. But if you had a few, 100,000 mammoths up across the tundra clearing all those trees off, it potentially could help reduce global temperatures by giving us a bigger area of permafrost that help keeps the planet cooler. It would sequester more carbon from our atmosphere. They could really help the planet. So there's some really good science behind the reasoning to want to bring these animals back, but it is a very different scenario to a Tassie tiger. 88 years gone. Anybody who's been to Tasmania will see that there's still absolutely pristine parts of that wilderness. All the animals it used to eat, in its diet are still thriving in Tasmania, out of balance for sure. They're all going a bit nuts, but there's certainly no shortage of food for a Tassie tiger to eat, and no shortage of an environment for it to really be able to thrive back in again. So it is a very easy, I think, demonstration of where we want to use de-extinction. And then other ones are a little bit different. Like the mammoth is really about this. Can we use these animals as sort of planet engineers to help us, again, undo some of the wrongs that we've done? The mammoth is a really cool one because mammoths are more closely related to Asian elephants than Asian elephants are to African elephants. So they are really similar. When you compare the DNA of a mammoth to an Asian elephant, it's like 99.9% or more identical. These are really incredibly similar. So they have a lot less DNA editing to do than we do on the Thylacine project. But then once you've got that mammoth cell, mammoth gestates in its mum for about 22 months to produce that, baby that's over 500 kilos. So that bit's going to be really challenging for them. Whereas for us, the dunnart, it's less than 2 weeks. It's 13 days that they have a pregnancy cycle for. And then, the baby's like 50 milligrams and it's about the size of a grain of rice. So that bit for us is easy. The engineering part's going to be quite a lot. It's going to take us a long time. And for the mammoth, the engineering's easy, but then having a surrogate actually, be able to raise, bring that baby up to birth is going to be very, very challenging. So we have different challenges, which makes the race more interesting, I guess.
Yeah, well, is it not a incubator option? I mean,
yeah. So for the marsupial, we could maybe culture them. For a mammoth, people are working on that. It's one of the things we've been looking into quite a bit, as well as just seeing how long could you do that. But nobody's been successful in growing, a human baby outside of a body, or a calf, or any of the livestock species. We're really still not very good at it. But
yep.
It's definitely a huge area of scientific endeavor, is trying to figure that out. There's obviously huge implications of it for preterm birth in humans, so lots of health implications for it, but also for livestock. If you could produce 100 cows in a factory as opposed to having pregnant cows all the time, or sheep or whatever, it would be an incredibly good option for feeding our growing human population.
Yeah, for sure.
Yeah.
So talking about populations is a great segue there. So how many would you need of the thylacine for a healthy population? And then would a smaller population have impacts such as like inbreeding and the like?
Yeah. So we have gone around museum collections all over the world to get all of their thylacine specimens. We've sequenced the DNA now from about 15 different animals, but we're going to do 50 by the time we get to the end of the project.
Yeah.
And we've got animals that were all shot as part of that bounty when they were hunting the Thylacine to extinction. So we've kind of got like the most contemporary or recent animals. So we've got about 20 of those. So we have a good idea of what their population differences looked like right when they went extinct. And then we've got animals that were collected 50 years or more before that, so when the population was much larger. And then we have animals from mainland Australia. So they went extinct on the mainland about 6,000 years ago. And so we've got some DNA from the specimens here as well. So we can go back very deep in time and bring back a lot of diversity in those animals. But I think because we know what all those differences look like, we can engineer 50 animals diversity, so all of the differences that exist in 50 animals, into maybe 10 animals' DNA. So we can make them really a good mixture of very, very different parts. So it would be like, bringing dingoes from the very northern part of Australia and mating them with dingoes from the very southern part of Australia. You've got ones that have evolved independently for a long time, and their genomes will look quite a bit different. That's what our first group of thylacines would be. And then they will produce offspring that have a mixture, because every time we reproduce, we give different genes to each of our offspring. That's why our kids look slightly different. And so when that happens, each one of them will inherit some of those differences, and that will build, we would hope, a really healthy population. And then we can go back further in time. So if that population's not thriving again in Tasmania, maybe we use some of those mainland animal differences and engineer those back in to make that population even more healthy. But we don't want to over-engineer it either. We want to make sure that thylacine that we recreate and put back into the landscape is that animal that was in balance with all of the other species there. So if we make it too healthy, if we put too many of those differences back in, we might then make it out of sync and make them too successful. There'd be too many thylacines, and then they could start having a problem of their own. So we'll go with, sort of the diversity they had when they were wiped out, and then take it gradually from there.
That just is like, how long's a piece of string? How do you get that fine mix and hope that it's not going to be this super predator that then its population gets out of control? But I mean, that probably wouldn't be the worst thing in the world, because if you have too many predators and their prey species runs out, well, then that's starvation, because there's not enough food, or they have less babies. And so it would sort of work its way out. You probably just don't want to see them dying off more than thriving.
Yeah. And we've certainly exterminated them once before, right?
Well, I mean, that's the backup plan, I suppose, you could have there.
No, but there's a great example of this. The best example in the world of this, actually, is from America in Yellowstone National Park. So in the 1930s, there was a lot of recreational activity, people camping, families and stuff in Yellowstone, and people were freaking out because there were large packs of wolves there and people didn't feel safe. And so they decided to eradicate the wolves from Yellowstone. And so there was, again, just like in Tasmania, very effective hunting program that completely wiped out the wolves from Yellowstone Valley. And then over the course of the next 60 years, they really watched that entire ecosystem, because this is such a well-studied national park. There's people there all the time that have been keeping population surveys of bison and deer and everything you can, bears, everything you can possibly imagine in that valley. They saw everything go off kilter, exactly as we're seeing in Tasmania today. So explosion of all of the prey species. So in Yellowstone, this was really deer. They just saw a massive explosion of deer. And then the deer became so abundant that the browsing pressure that they were placing on the trees and the shrubs there was extreme, was something that had never been seen in Yellowstone before. So there were certain tree species and shrub species that just failed to grow at all, because every time they shot a new shoot, there was a deer there chewing on it and doing it. That stopped all these young trees from growing. And then the beavers had no trees alongside the river to start, chewing down to build their dams. And so the beavers started to move away from the area, and it actually caused the Yellowstone River that runs through Yellowstone Valley to change its course. It actually changed direction completely because of the lack of these beaver dams. The deer population exploded. The landscape got completely decimated. They were, having landslides and all sorts of stuff because the shrubs were going. It was really terrible. And then they realized this, in the mid-1990s. They were going, this all seems to stem from when we eradicated the wolves. And so they launched a, very forward-thinking program then to reintroduce wolves back into Yellowstone Valley. So fortunately, they had populations of wolves in other national parks around America that they could get animals from and put them back to Yellowstone. We don't have that luxury in Tasmania. And then over the course of that last 30 years since they went back, that entire ecosystem has rebalanced itself. There were wildflowers and shrubs and trees that grew for the first time in 60 years as soon as the wolves were brought back. The beavers came back into the valley because the trees were back alongside the river. The river regained its course back through the valley. And it's just this incredible example that I think as scientists and ecologists, we often think about, if you kill the thylacine, then all the prey species are going to go crazy. And we definitely see that. But with this beautiful example at Yellowstone, you can see that actually it impacts the trees, it impacts the shrubs, it impacts the bloody river and how a river ran through the landscape. Like, it impacts things in a way that is so complex, we can't even begin to comprehend how important those animals are in that landscape. And that's why, we know the wheels are beginning to come off in Tasmania. And unless we can get that predator back there, we will continue to see this ecosystem falling apart. And we can try and manage it ourselves. You can have people go out and shoot the deer in Yellowstone Valley, but it never brings everything back into balance the same way of having the species there that actually kept things in check does. That really brings things back into a healthy balance. We can attempt to do that, but we never really achieve it.
I think that's probably, that domino effect is just unbelievable, and it has such far-reaching complications that we can sort of see. And Yellowstone is just that perfect example.
Yeah.
And that's been fascinating to see how that's played out, and the recovery that it's made is just amazing. And do you think because the thylacine in Tasmania was sort of the one-off, whereas on mainland Australia, we've got, the dingo. So it's sort of that, diversity in an apex predator that what we're seeing has been amplified in Tasmania because they were it. And with them gone is just, it's like a free-for-all.
Yeah, it's exactly what it is. There's no other animal there to kill things. Here on mainland, we've got dingoes, we've got foxes. We have a lot of issues with introduced species here. The dingo not being an introduced species, it definitely came into Australia a long time ago, but I think it's fair to say it's probably a native species at this point. Like, it's
by this stage,
Yeah, right. It's very much in balance with the rest of the ecosystem. But, even at Wilson's Prom here in Victoria, they were talking about some of the imbalance they have in marsupial species there, and that they were thinking of trying to bring dingoes down into that area to actually have an apex predator back there, because we don't have one. And so you see explosions of wombat populations and all sorts of things, again, going out of balance, huge explosions of wallaby populations, and just recognizing that, we probably need an apex predator back in that landscape to help bring all those animals back into check.
So with the thylacine, we're getting hopefully within that 5 to 10 year bracket that you've done all the sequencing, you're all good on the DNA side of things. How quickly could you get that population up? Because with the surrogate mothers and things like that, is it something that you could do 50 at a time? Is it a bit more complex and a little less numbers, or is there a scale up procedure for that?
We hope that we could rear, a lot of them at once, maybe 50 at a time. That would be amazing. We're trying to figure that out for just rewilding our own, living marsupials after bushfires and things like that. Like, it would be amazing if we could create 100 koalas, either using surrogates or growing them in the lab and then being able to re-release them after areas have been burnt out following bushfires and or floods, any of the horrific weather events that we seem to have so regularly in Australia now. They're really wreaking havoc with our native animals. And so, some of those things would be great to be able to do. But that would be the initial thing. We would kind of ramp up numbers, but then they would be bred in, very large tracts of land in Tasmania on that landscape. And you will have to study them for a good long time to make sure that they're having a positive impact on that ecosystem, that they're not going to cause any, untoward problems. God knows, as humans, we've introduced enough species and caused enough ecological disasters that this is something you would have to do in a series of stages of releasing them into bigger and bigger areas and really studying exactly what that interaction is and making sure they're truly bringing everything back into balance. One of the things we know that's a pressure in Tasmania now that never used to be is wild cats. So there's a lot of wild cats across that Tasmanian landscape now. And so putting the thylacine back there, you're introducing another predator when you've got cats in that landscape. Obviously, cats are not taking out wallabies and the larger prey, so they're no good in that apex predator role. They're killing the birds and all the small marsupials and the things, that are really endangered, which is awful. Wild cats are absolutely awful. And so, but one of the good things is that we know cats hate other apex predators being around. So if you have a fox in an area or a dingo, cats will not go into that area because they're a prey species for those larger apex predators. And cats are very smart and they know just to keep out of the way of bigger apex predatory animals. And so one of the great things would be if we were to put thylacines back into the landscape and they move into those really dense bush areas of Tasmania, we would love the idea that hopefully they push cats out of those areas and get them into more open, exposed areas where they're going to do less damage. And that helps protect those forests even more. Obviously, thylacine can't climb a tree and, kill birds. It's not going to be able to get into tiny little burrows and kill some of those really small marsupial species. It's going to be, like we said, much more in balance with that whole ecosystem. And there's some other really interesting things we looked at. So one of the things that they showed, again, in America, where they've done a lot of these studies, is they eradicated mountain lions from California. And then again, many years later, they brought mountain lions back into some of the mountain areas in California. And one of the things that they noticed, to their great surprise, was even though mountain lions have been gone for over 100 years, the animals there seem to be able to recognize mountain lions and see them as being a formidable, apex predator, and immediately evaded that species. And so there was a lot of concern initially that because none of the animals on that landscape had ever seen a mountain lion or been raised by parents that had run away from mountain lions, that they would just sit there and get eaten by them. But instead they were actually able to go like, this looks like a scary animal, I'm out of here. And so we were kind of following up on the theory that there is this sort of genetic memory of a predator in a landscape, that animals, it is imprinted in their brain. And it makes sense, right? If you're an animal raised on a landscape that is a prey species, one of the most important things for your brain to be able to do is comprehend a risk and to avoid that risk, because basically you spend your whole life just trying to not be eaten. And so we got replicas of thylacines made, very realistic replicas, and we went out into the Tasmanian bush and we put them out, in cleared areas, and then we track what happens when marsupials come out to graze on those areas at night. So we have a replica of our thylacine, a replica of a goat, a replica of a fox, and we have nothing. So we have the absence of it, just have a camera stuck in the middle. And then we measure what the animal's responses are to those different replicas. And you can see without any shadow of a doubt, the animals definitely recognize the form and the striping of a thylacine, and they are not hanging around that animal, whereas they disturbingly don't care too much about a fox. And then the goat, is obviously not a threatening species, but they're animals they're quite happy to sort of just graze in front of. But if there's a thylacine there, they do not like being close to that thylacine. They'll be in the distance and keep an eye on it. You can actually see their little eyes on the night vision camera kind of looking at the thylacine, but they won't come in too close. So that's another good thing, is there still seems to be that imprinted memory in the prey species in Tasmania that when we do release thylacines again one day, we're not putting it into a naive environment where none of those animals know that this is going to eat them. They're actually going to be pretty good at recognizing that that's something they should try and run away from.
That just amazes me about animals, that ability to pass that on.
Yeah.
And like, they're not like humans. Like a lot of animals don't raise their young and teach them the way. I know there are species that do, like dingoes and things like that, but for a lot that it's just that genetic makeup and it's just instilled into them, it's imprinted into them. That is fascinating. So that is amazing that would happen to those animals already. And it's just in their nature to know, that we need to worry about.
Yeah.
How hard is this from a government side, and like not so much grants, but like sort of the legislation and logistics? I'm guessing that's got to be a pretty big undertaking, that once you've done all this work and got it to the point where you're confident, say, hey, we can do this.
Yeah.
Then who controls it? How does it play out? That must be a big thing that you guys, are you thinking about that yet?
Oh, yeah. So we're working on that now, because it probably will take us as long as it will take to make a thylacine to start getting some really good conversations around this. It's really hard because there's still, we're 10 years off at the moment with today's technology having a thylacine. And so if you want government to really start seriously considering what they'll do with these animals, it just seems that's way too far away for them, right? Like, it's like, so in the future, they're like, we'll worry about it when we've got a thylacine. But projects like the Northern Quoll, where we're changing that one bit of DNA, and it's a way that we can do genetic engineering to save a wildlife species, that will then be the areas that we start to then really push policy around what we want to do with these animals, how we classify these animals. At the moment, when you genetically engineer an animal that's considered a genetically modified organism, or a GMO, which is, a term you've all heard of before. We hear about GMOs all the time. And we do have regulations around GMOs. In Australia, we're pretty relaxed around GMOs. There's only real issues around them if you want to eat them. Hopefully people do not want to eat thylacines. And so, it would be considered a GMO. But then as a GMO, it wouldn't get a wildlife protection status or anything like that, for example. But because this is the first time that people are really pushing these technologies, and these are discussions happening globally, so it's not just within Australia. There are a lot of big groups coming together now, really talking about synthetic biology, how that's changing not just wildlife and conservation, but our whole existence, right? Because we're talking about editing out human diseases, because we do have that technology. So for things like cystic fibrosis, where we really understand the genetic basis, we can use this technology to remove that from somebody's DNA, so they're not going to pass that on to their children. Like, we have the ability to do these things. And all of these come back to, where do we want to use this technology thoughtfully? And I would hope for projects like Thylacine De-extinction, that's one of the areas that we can all agree this would be a really great thing to be able to do for the landscape. Tasmania is our last stronghold of so many marsupial species that if we're going to try and save any ecosystem, that's one of our most important ones. And that putting that apex predator in there will really help. But when also do we not want to bring things back? Like, we can't do dinosaurs. People always ask me about dinosaurs. No DNA left in them. It actually breaks down pretty quickly. So, there's certainly nothing left in a 60 million year old sample. Jurassic Park got it wrong. No amber mosquito DNA thing. But, you can go back as far as you can get DNA. But like you raised before with the mammoth, it's been gone for a really long time. The world's quite different now. Is that something we find as palatable as bringing back a thylacine?
Yeah.
And then what are our reasons for bringing it back? And then, and then really addressing those sorts of questions.
Yeah, the whole thing is just absolutely fascinating for me. I just sit here and you have one of the coolest gigs going. And I know
I do love my job.
Oh, I don't doubt it, because I think in 10 years' time, when this plays out, it must be it will be a pretty amazing accomplishment. But just to say that you spearheaded this, and that you've, from inception, and a life's work to get this happening, is just unbelievably cool. So, mate, I really appreciate you coming on the podcast and telling us about it. And I cannot wait for the day that we see a thylacine back on the landscape due to the hard work that you and your team are doing.
Me either. I cannot wait for that day.
So,
Yeah.
well,
Awesome.
mate, I've had a blast. I wish you all the best. Hopefully it won't be 10 years. Hopefully.
Yeah, I hope not.
It will be, but closer to the 5 would be nice. And I'm sure with the advancements in technology, they seem to be jumping ahead.
It is
very
jumping really quickly. So yes, I have great hopes that it will be a shorter time frame.
Yeah. And I mean, it must be pretty cool too, jet setting around the globe, dealing with just other countries and their, just the academics that you must get to liaise with and talk to. And that just must be unreal.
Yeah, it is a really great gig, I have to say. I'm a very fortunate scientist, so I'm having a great time.
Well, mate, congrats. And I wish you all the best, you and the team there, and can't wait for this to all play out. So again, really appreciate your time tonight on the podcast. Guys, if you haven't, Google Tigrr Labs, and it is Tigrr Labs. You'll go straight to it. Some fascinating stuff. Can people help out and donate as well?
Yeah, there is actually a donate button on there. So if people want to get involved, we have a little bit of merch as well that we send our donors. So if people give us a donation, you get a bit of Tiger merchandise as well. But we really love having conversations with people. If people have questions about it, comments, I'm really happy to answer people's questions about this science. So, I'm really all for educating Australians in particular around why de-extinction science is actually a really good thing and something we should all get behind.
100%. Great way to end there, guys. Check it out. Donate. I mean, that's pretty cool to say you donated to bringing back the thylacine. This is, let's hope the Aussies do it and we get the first one there. So, yeah. All right, Andrew, appreciate your time, and thanks, everybody listening. Bye for now.